So welcome all to the 11th edition of the Ramanujam Math and IT conference. This has been co-hosted with the weekly Samvaad series. So to be precise, this is going to be a virtual mini conference featuring talks by two wonderful speakers. So this conference, RMIT, is aimed at celebrating the genius of our nation's great mathematician Srinivasa Ramanujan and attempts to bring together researchers in mathematics, information theory, and other applied areas of mathematics. So we have with us Professor Vijay Chandru. So I mean, for formality, let me introduce him. So he's an academic and an entrepreneur. He's a co-founder and director of Strand Life Sciences. He also serves as an adjunct faculty member of Biosystems Science and Engineering and a visiting professor of the Robert Bosch Centre for Cyberphysical Systems at IISc. He got his Ph.D. In operations research from MIT in 1982. He has taught and conducted research in the computational mathematics of optimization, geometry, logic, and biology at Purdue University and the Indian Institute of Science. He has co-authored a book in computational logic, edited several volumes, and published over 70 peer-reviewed research papers. He was elected a Fellow of the Indian Academy of Sciences in 1996. So Chandru was awarded the Hari Om Trust Award by MHRD in 2003, the President's Medal of Informs in 2006, and was named in the 50 Pioneers of Change by India Today in 2008. So we had Professor Chandru's lecture at the last edition, and we are very happy to see him again. And he'll be speaking on computational epidemiology and COVID-19, which is a very apt talk in the current scenario. And before that, I would also like to welcome the students of the epidemiology course he teaches us at IISc, if I'm not wrong.
Yeah.
So over to Professor Chandru.
Thank you so much for that introduction, Srinivas. I'm very happy to be part of the Ramanujam celebrations again, but obviously, we couldn't be at the campus of IIIT, but yeah, nevertheless, good to participate. I think this COVID-19, of course, in some sense, has been a major event in our lives, and for our if you look back in history, there are these monster kind of events that happen periodically, and every time there is an associated sort of social turmoil that happens. And this time is no different. I think there's a lot of challenges from a social perspective. But I think what has been uniformly clear is that from a scientific perspective, we have never been as well prepared as we were for this one. And possibly the lessons learned from this one will make us even better prepared the next time around. From a mathematical and computational and biotechnology perspective, how to fix the social problems, I don't know. So I don't think I have insights on something like that, but I can tell you a little bit of how the mathematicians and informaticians have responded to the crisis, and I think that has been very heartwarming, that we've been able to effectively assist in probably mitigating some of the effects of this pandemic, right? The work I'll describe is still developing, so it's still in its early stages. So I hope you will forgive me. There are no nice, crisp mathematics that can be laid out to describe all of it, but it is exciting because it's sort of ongoing work, and I've been fortunate to now collaborate with a few uh, very creative people on the biology side, Charles Cantor, um, from the informatics side, a number of different people, including Manoj Gopalkrishnan, a very bright young faculty member at at IIT Bombay, um, Rajesh Sundaresan at IISc, and then of course Bud Mishra and others from NYU. So Inavamsi is now at IISc, but he was a student of Bud Mishra's at Courant Institute. Um, so there we go. So epidemiology again. I'm sure most of you are familiar with this word, particularly in the context of the pandemic. We keep seeing this mentioned, um, and and the word itself, of course, comes from the study of health and disease in populations, from its origins of epi and demos, and historically this goes back to the origins of medicine, healthcare, Hippocrates, and so forth. Um, and you know, it's a very classical discipline in public health, and in India, ICMR actually runs an institute for epidemiology in Chennai. And very early on in the outbreak of COVID, there was a model and a predictive model that was published in the Indian Journal of Medical Research by that group from ICMR, and which led to some decisions like the lockdown and so on. But we'll talk a little bit about that now. What the model that was used was really a very classical epidemiological model, which when you think about it, it's actually quite a simple model. There are the population is put into three compartments: either you're susceptible, or you're infected, or you've recovered, right? And so that's why these are called SIR models. And so there is a rate at which susceptible become infected, a rate at which the infected recover, and so these rates essentially describe the dynamics of the of the epidemic, right? And these the transmission rates and the uh, infection rates I mean, and the recovery rates are modeled by these beta and gamma. And then there is a number that is called the reproductive number, the R0. And it is the R0 which sort of determines whether the epidemic grows or it dies out, right? And so the big focus is always on that number. Is it greater than one? Is it larger than two? And and those values will determine how rapidly the infection grows. Um, so as you can see, when the effect of the R0 on the epidemics, on the left you see the curves that you get the very steep vertical curve when R0 goes to about 3 6.0, and then it slowly flattens. So you would have often heard flattening the curve, right? And how you flatten the curve is by things like lockdowns or masks or social distancing and so forth, so that you bring down the infection rates or the transmission rates, and that slows down the spread of the infection. And so you can actually use these models to study the dynamics. In fact, early on, the modeling approach was they took the four uh, sort of metros, north, south, east, west, of India, and looked at the air traffic uh, between these cities, and on those assumptions, they actually modeled using just these SIR models how the infection was going to spread, and I think based on that, some decisions were made strategically on the lockdown, etc. Right. So this is a very classical biostatistical modeling methodology that epidemiologists have used for almost 100 years now, right? Now, the limitations of these compartment models has been seen quite often because, in some sense, we don't live in compartments, right? And we don't actually interactions between uh, members of society and so on are quite complex, and so these models might not really capture what happens, and and they've been off in some cases. For example, the earlier SARS outbreak, which happened almost 20 years ago, they were really worried because they thought the R0 was going to be, somewhere very high, 2.2 to 3.6. And this was an infection that was spreading uh, like what we are seeing with COVID, but it was, their measurements were all based on hospitals, and the measurements were based on how the transmission was happening within these very closed environments of hospitals, and and where they thought that the R0 was a fairly high number, and finally, the outbreak actually got controlled because the R0 was really not that high, and um, so ever since then, I think epidemiologists have been looking more to how to build more high fidelity models using agent based simulations, and where you actually also, from compartment models, you move to actual graph representations and network representations of how people interact and how contact can happen, and then you run these large agent-based simulation um, models to get better predictions and more fidelity into the models. So this is um, just to give you a sense of how far you can go with this type of modeling. In March, end of March or so, a group at IISc started modeling the population of the city of Bangalore, right? This was a group led by Rajesh Sundaresan at the Cisco Center for Network Intelligence at IISc. And the group actually built a model of 13 million agents, which was roughly the population of Bangalore city, and actually modeled the 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 cities, geographical dispersion of the population, and they also had data on um, you know schools, workplaces, and movement of population and so on. And the same model was then adapted for Mumbai, and I think a group at TIFR led by Juneja, also adapted the model for Mumbai. So, and these models were used in some sense to uh, both for tactical and strategic planning of responses to the pandemic. Uh, the but these models are big. They're and they require a lot of computing power to to run the simulations and get the predictions. So there was a lot of data science that had to be brought in to actually create these models, and that has become kind of the modern way in which epidemiology works, and that's why it's starting to be called more computational epidemiology or digital epidemiology, as the course at IISc that I teach is named. And this um, of course, in the US, it's been used quite a bit for modeling the flu trends, and of course, Google made a big impact with their approach called Google Flu Trends, right? But there are many groups now that now use a lot of the data science techniques um, to uh, to model these epidemics, which are seasonal epidemics, of course, in the US. And in fact, a couple of our former students from IISc, Anil Vulikanti and Srini Venkatraman, are leaders in this type of work. They sit in a very exciting center at the University of Virginia. They're all faculty there, and the senior professor Madhav Marathe, who's probably Sadgopan might know him from Kanpur. He's an IIT Kanpur.
Yes, of course. Yeah.
Yeah, yeah, very good. And Madhav's, of course, been a superstar in this whole business. So, in fact, all of these people used to be at Los Alamos. They used to work on epidemiology in biodefense, right? Because this was always a fear that epidemics and pandemics could be created by biological warfare, and but then they decided to switch from biodefense to looking at public health. So that was then they moved to the university to actually build something that was more public health oriented. And this group maintains, some of the best tools and platforms, and many of them are accessible. And they just recently moved from Virginia Tech to University of Virginia, so I'm not sure if they brought up all the systems um, at the University of Virginia, but they're the group to track on this interface of epidemiology and data science. Incidentally, Anil Vulikanti was my PhD student, so that's how I got interested in all this, and I've been sort of tracking this. So Ramesh Hariharan and I were his PhD guides. So this happened many years ago at IISc. So let's actually now start talking a little bit about COVID itself. And without uh, before we jump into that, I do want to point out that this time around, we've had a couple of interesting advantages. And as I said right at the beginning, I think our ability to analyze the virus and understand how it works and what its genetic code is, and sequence it and study the variants of how it's mutating and so forth. A lot of that, we were so quick to do that with this epidemic. And the reason is that genomics has come a long way, just as Moore's law has improved computation significantly. The law of genome sequencing, which is sometimes called Flatley's law, named after the chairman of Illumina, um, is is also, an exponential law that has rapidly brought down the cost of sequencing, and also made it much faster and much easier to pull off. That's on the one hand the ability with genomics, and on the other hand, of course, there's been lots of advances in digital health and in general. And so the question was, how did we, cities like Bangalore, Science City, and so on, respond when these crises, when the crisis hit us? And as I was pointing out, some of the modeling work that Rajesh Sundaresan and group started at IISc, and but then there's also some very nice work that was happening on understanding the genome itself. As you may have read, the genome was sequenced, pretty much within a few weeks of the crisis being announced. Right, by about the end of January, the sequence of the virus was published, and you understood that quite a bit about the structure, and the actual coding regions of the genome of the virus, and what they meant, and why, and how you could actually leverage that information to very quickly come up with tests, for example, the RT-PCR tests and so on, which actually needed this sequence information. Could the work on that started up right away, because as soon as they knew what the sequence was. And of course, there have been various depictions of this so-called spike structure, and which in a way has become the picture of one. We as humans, we really need a visual representation of the monster, right? The beast. And so this is how we all see it. And probably the hand-drawn sketch on the right, which is from this wonderful new book that has come out in MIT press. I think I had it on the previous slide. Yeah, Arup Chakravarti and Andre Shaw. With all these illustrations by Philip Stork. So this illustration, the coronavirus, is from that book, and yeah. So so what more could you do once you had this sequence, the reference sequence of the virus, is that you could actually then start getting samples of patients who had been infected across the, in our case, in this diagram represents work done by Chitra Pattabhiraman at NIMHANS, who's probably one of the earliest virologists in India to work on metagenomics of viruses. She did extraordinary work at NCBS as a PhD student when she started diagnosing dengue strains across India using metagenomic sequencing techniques and sequence analysis, basically, of where there could be multiple sort of pathogens and DNA, viral DNA and RNA that would need to be sorted out, right? In the case of SARS-CoV-2, and I think this particular data is based on about 94 samples or so from across the state. She's probably done a lot more now. And basically she identified various clades, as they're called, or, sort of small variations of the original virus, which in some sense could be traced back to essentially international travelers who brought the virus from different parts of the world, right? So there were people who were traveling back from China, people who were traveling back from Europe, people who were traveling from Indonesia, and so on, different parts of the globe that where the virus had taken slightly different shape. And so she was able to actually use this genomic information to actually find that certain variants had taken root in different districts of the state, right? And it happened, and it happened obviously because somebody brought that strain and then traveled home to that district, and then the infection started from that strain that was brought in and spread within that region. And so this is a slightly different way of understanding the epidemiology of the disease, because now you're actually using viral identification or sequence identification, genomic identification to see the source of infection and how it has spread, right? And of course, eventually these clades will probably mix and keep evolving. So that will happen, but at least there was some early tracing of how the infection spread in the state of Karnataka because of her work. And the fact was, NIMHANS was a major center for sequencing samples that were coming from all over the state. Chitra also had a sequencing lab set up in the same premises, so they were able to essentially carry out a research study alongside the actual clinical diagnostics work that was going on. And this has been the kind of scientific understanding and breakthroughs that have been happening right through this COVID pandemic, and I think it's as I was saying again, the science has been immaculate. I think in trying to very quickly get to an understanding of the virus, the disease, and move towards therapies and vaccines and so on at an incredible speed compared to what happened, say, with HIV or any of the earlier sort of epidemics, right? So I think I hope that was the message I wanted to convey with Chitra's work. Her preprints are there on medRxiv, and some of them may have already appeared. And she's also been writing very nice blogs on the COVID Gyan portal, which is maintained, I think, by ICTS as a public service, right? So I wanted to spend a little bit of time talking about this idea of biomimicry and how this relates to this virus as well, and how, in some sense, we'll need to understand that there's also a level of gaming that the virus does with our immune system, and we'll need to model that better if we want the vaccines and so on to work. There is no guarantee right now that the vaccines are going to work, right? I mean, we're still carrying out the trials, and these trials have to go on for a long time before you actually understand how these work. But here's an interesting image from this paper by Stephen Massey and Bud Mishra, which appeared in the Journal of Royal Society, where they described the various types of mimicry that you would see. There's deception in signaling. So there's a beetle that actually puts on these yellow stripes to sort of deceive the predator into thinking that it's a wasp and not a beetle, right? And and that's called Batesian mimicry. And then you have these Mullerian mimicries, where essentially organisms will try to form collectives for protection. There's now, the interesting thing about this biomimicry is that it can happen at a molecular level. So you can actually study it at the level of proteins and signaling that is happening within say, a cellular system. You can think of it at an organism level. So while we've been talking about beetles and wasps and so on, and from a game theoretic point of view, you can also talk about it at a social level or sort of behavioral level. So deception, can also be, asymptotic patients who will essentially tell the system that they're not spreaders of the infection, because they don't show any symptoms, right? And so they can get away with that deception, right? So they might have been in a high-risk situation, but because they're asymptomatic, they don't, you know, they're allowed into various high density population places, while they obviously know that they've been at risk somewhere, but they can deceive the system because they don't have fever. So those infrared guns which are checking whether or not you have a fever, they don't have a a breathing issue or coughing, and so again there's a certain amount of deception that can happen because of that. So as I was saying, I think socially this is a very complex epidemic because because of issues like this, right? And then there are various types of mimicries. There's mimicry of, for example, again, like wasps and bees will form a ring so that they look like a much larger organism, and that fools a predator that might be looking to pick up an insect to eat, right? So, but when it sees this this swarm, it imagines that it's a much bigger organism, and then you have things like Q mimicry, which is the classical chameleon, right, of changing colors to blend into the environment. But the funny thing is, you can also see that with at a cellular level, where cancer cells can hide in tissue by certain masking using, I think there's a chemistry, I think salicylic acid or something like that is used for this antigen masking, and they basically, the tissue looks like it's normal tissue and it's not cancerous, but it's basically the cancer cell, playing games, and so. And then it goes on, right? You can look at complexity and stability, and then what is called costly signaling, right? Where at a molecular level, the structure of the protein sort of is the basis of the signal. With animals, it might be sexual ornamentation. And at a social level, it could be, health workers, taking unnecessary risks, right? Which is an expensive proposition, because I think at least in the early stages of the infection, maybe many of the health workers didn't understand why it was so important to be with protective gear, and many of them exposed themselves, and many of them obviously had very serious infection, because this particular infection seems to be largely dependent on, or I mean, one of the explanatory variables for people who get very sick is the amount of inoculum that you're exposed to in the infection. So the blast of the virus that you get is that gets the infection going in you is actually a good marker of how sick you will get, right? So, and that's what the health workers are really at risk for, because in a health in a hospital or in an environment where there are many patients and so on, this can get to be a serious problem. And that's why you need all those PPEs and so on. So it turns out that there is if you look at the SARS-CoV-2 virus itself, it again uses mimicry of various kinds, and this the analysis of that has been presented recently by the same guys, Massey and Bud Mishra, but this time joined by a game theorist, William Casey, and I've been just reading their preprint on this, and it's again, a use of mimicry at a molecular level that is see, furin is a particular enzyme that needs to cleave proteins to make them function, right? And so essentially the SARS-CoV-2 virus has various ways of getting furin to work on the virus. So I think it's this is on the spike protein cleavage sites. So it's this is starting to give us some understanding, but I think it does suggest that we're going to have a little bit of challenge as the vaccines, how effective they're going to be against this virus. Right? Go along. The mimicry. Oh, there's a little illustration there about how mimicry, at a sort of social level, is used to deceive, a customer into thinking that they're getting a nice bottle of Chianti wine, but these are actually fake wines of different types, right? And you can see how physically they can mimic. So finally, I think I want to point out that that this is this COVID, it comes under the class of strategy problems that are were classified in the 70s at Berkeley by people in urban planning and so on as wicked problems, right? And Rittel and Weber, I think, yeah. So that's the reference. But more recently, I think in business strategy, the Harvard Business School and so on has a number of papers on in the review HBR on wicked problems and strategies for addressing them. And the description or the definition of a wicked problem is listed there. In fact, it starts out by saying that there is no definition, there's no definitive formulation of a wicked problem. But they have certain characteristics. They have no stopping rule. They solutions are, this is not like a math problem where you either have a true solution or a false one, but here you have to choose between good and bad, and there is, there's no immediate or no ultimate test of a solution even, and so forth, right? So, and the worst part is, of course, the planner has no right to be wrong because lives depend on it, and it can have devastating effects, right? So, so this is a classical sort of wicked problem, and there are only a few ways in which you can approach wicked problems, and we've seen these being used in the context of COVID. Now, which is the most effective, we'll not know, but until this whole thing dies down. But there's the authoritative, there's the competitive, there's the collaborative, and so forth. Clearly, the collaborative, you could see that Scandinavian countries like Sweden and so on have been able to pull off something that is slightly, that was very interesting in the beginning, right? Because they didn't have any lockdowns or anything like that, but they were very collaborative in social distancing and masking and so on. And whereas in other situations, they were welding doors shut so that people couldn't move around, and so clearly authoritative action and so on. Yeah. And in some ways, the severe lockdown that was instituted in the country initially had that authoritative flavor, but it, that I don't know if it was entirely effective, but it was of that kind. So one of the things that we are trying to do now as a group is to start trying to build towards decision supports that can use mathematical modeling or simulations to start looking at different protocols and so on for that might help with keeping social units as safe as possible, right? And so we're looking at this more from the point of view of a campus or a gated community or schools, which where you could actually try out different strategies and come up with decision rules that could or protocols that could keep people safe. And this is the work but the group that I mentioned on the title slide, we're all collaborating on this, and it will and we've given ourselves a name. It's called the Corollary Health Group, and we're trying to build a framework that can come up with various badging protocols, maybe time slots, saying that there's certain time slots in the day that a person with a certain type of badge can enter, so the times you can go to the library, the time slots for going to the mess, and so on, to actually control infection spread. And similarly, quarantine strategies for different areas of the campus and so on. And I'm sure, campuses that are reopening, and it looks like the month of November, we're going to start seeing a lot of campuses reopening, are going to adopt strategies like this. But it would be nice for them to have a tool, decision support platform that they can use to decide what rules make sense and what don't, right? So that's a platform that we're in the process of building. So I also wanted to just mention very briefly a few very nice pieces of work been done on ideas of, for example, testing, right? Suppose instead of having to test everyone, you can actually do pool testing. So you bring down the cost of testing, and so that if you pooled 20 samples and it came out negative, then all 20 are negative. So you get a great reduction of cost there. Of course, when there's a positive case mixed in the 20, then the question of how you resolve that is is a little more complicated. But there's a very nice structure based on Steiner triples that Manoj Gopal Krishnan and team at IIT Bombay, and along with some physicists at NCBS, Sandeep Krishna and others, they figured out a scheme called tapestry pooling, where you can actually pool samples, and the same individual sample might go into multiple pools. It'll actually go into exactly 3 pools. I mean, that diagram shows 2, but typically will be exactly 3 pools into which each sample goes. And then when you get the output of the pool samples, then you can invert the linear system. It's a sparse linear system, so you can actually get back the unique solution, and you will know exactly who is infected and who is not. And you'd also know the level of infection because it's a quantitative solution that you can work out. So this method is probably one of the most clever methods that's been invented, and can actually get you a pretty good reduction in the cost of testing. So this has all been now validated with Tata Memorial Hospital, Malabar Hospital, and others. And I think the papers have now gone to ICMR for approval, but it's a very clever idea that actually brings together some nice mathematics, some computer science, and coding theory type of use of methodologies in this is called compressed sensing, right? And group testing, and this is one result that has come out. Yogesh Simhan and Tarun Ramba and others at IISc have built this thing called Go Corona Go, which is a contact tracing app. And this is now on Google Play Store and Apple Store. And we have supported it through the DST Rakshak program. I'm on the coordination committee, and we've and now it's been deployed at IISc and IIT Jodhpur and IIIT Hyderabad. So it looks like this will be, and it's nice. It's because it's privacy preserving. It's well done from a back end perspective. Also, it's well engineered. And I think close to about 1,000 users now on IISc campus. So this is using Bluetooth, that the usual, like the Arogya Setu thing. So I think what we're hoping is that in a campus type situation, we will do a lot of screening using things like contact tracing and perhaps pool testing and so on, and various zero surveillance methodologies to advise the decision rules. Right. So this is once you get into testing, then you also get into regulatory issues, because it's a diagnostic protocol, and then you have to also be very, very careful, and there's lots of challenges with doing that because it's a clinical outcome there. I won't get into all this, but there are various now testing methodologies that are available, and many indigenous ones. And in fact, now Rockefeller University, Rockefeller Foundation has given a big grant to the Principal Scientific Advisor's Office. And that grant has been used to set up something called indigenous diagnostics. It's a marketplace that is being created at CCAM and by TCS. And so we will, the idea is to build procurement and the logistics of reagents and test supplies and so on, that will be done through this marketplace. And hopefully we are going to use all of this to make our campuses at least safe and allow. Of course, we will always have to reflect to state health and ICMR guidelines for clinical actions, but we should, there are many things that can be done to make sure that the bad clinical outcomes are few and far between, right? So this is again part of this Department of Science and Technology Rakshak program that where this is one of the projects. So there's a team at IISc and there's a team at IIT Kharagpur that are building sort of independent approaches, but we are sort of collaborating to make sure that we don't unnecessarily compete, but we'll collaborate in building these decision support frameworks. So I'll just end with that. There's lots of fun things you can do in studying epidemiology, and this was one of our students at IISc who used a kind of deep learning methodology using Gaussian processes to be able to build predictive models of dengue. She was in the first group of students who took this course that I teach and so on. I just want to mention that a lot of the work done at IISc on modeling, on trying, sound acoustics for COVID screening, you just on the telephone, if you hear the person coughing, then can you analyze that audio signal? There's a system called Koswara that has been developed by Professor Ganapathy and group and so forth, right? So there are a whole bunch of these. And what we have done is created some podcasts. So if you go to the IISc website, you can find these podcasts and, they're fun to listen to, and they're with these teams that have been working on these different methodologies. So with that, maybe I'll stop.
Thank you, Professor Chandru, for a very nice talk, and good to know that a lot of interesting, exciting work has been happening at IISc on this topic. Right. And also that COVID-19 is a wicked social problem, I think nicely summarizes what to expect next in the current pandemic.
Right. Yeah. So I think, there are some whole class of problems that that will now open up, because once the vaccines come, then what is your vaccination strategy? How are you going to pick the right populations to vaccinate and so forth, right? I think there's again a whole class of problems that will open up for that. Again, it's an area of epidemiology that has been classically studied.
Nobody has designed a vaccine this fast, and till you get it or whatever its equivalent, it's going to be difficult. But there are so many different platforms, and they actually work in very different ways and have to be handled in very different ways. What is the end for coronaviruses? So far, there has been no really strong vaccine developed. I mean, there's knowledge system, knowledge has grown from SARS and MERS, but but there's been no commercially licensed vaccine which has been approved, maybe because the disease itself did not spread too much and then the interest went down. But what is the current consensus on the likelihood? Of course, there's a huge amount of hype on all these things, but scientifically, what's your opinion on this?
I think we're probably going to see, some vaccines, conclude at least on the basis of a new type of phase 3 trial, which is a limited phase 3 trial, that they can probably go into the general population by, looks like March, April or so. We should be seeing some of that happening. It could happen sooner, but those would be, tricky decisions. But the bigger challenge is, what will you do when you start seeing some adverse events? Will you just, completely pull back? Will you just keep going? Which are the populations, on healthy populations, that you would try this on? Obviously, there will be some pressure to do it on the vulnerable populations, but then you may have some adverse effects, and then what will you do? And so I think this is just going to play out, and so I can't see this being, fully given to the entire population in less than maybe 1 to 2 years, right? So I think we need to learn how to live with this beast, right? And
so I think, I agree in that. The system is so non-linear. It's very hard to predict what will happen in any particular set of individuals, even given their medical history.
Right.
The standard vaccine procedure is way too slow, I think, for anything like this.
Right. Yeah. And but the other complication here is that we've not been able to fully, obviously, understand the complexity of the immune responses, right? And so that's where I think a lot of the research will probably go in also rapidly as these vaccines play out. They'll have to, alongside, be doing, they're probably doing that now as part of the trials and so on, but that will become much more.
Yeah, just one last question. You mentioned so many types of biomimicry. SARS does all of the COVID does all of these?
Quite a few of them, 5 different types of biomimicry. I think 5 different instances have been noted, right, that I've seen so far. It's I guess that's one of the reasons why people sometimes get into this conspiracy theories, that it must be engineered and so on, right? That it's because it's pretty clever in that sense. But nature is clever. Yeah.
So any other question? Please unmute yourself and you can ask the question.
Yeah, I had a question.
Please.
There was some in IEEE Spectrum. There was an article on using in identifying protein by doing genetic analysis on the genome sequence of SARS-COVID-19, identifying groups of drugs which can be targeted.
Yes.
But nothing. I did not see any follow-up article on that. So somebody who has already acquired COVID and is infected, and suppose he wants to, what is the treatment? So we keep on hearing about new drugs, nitrous oxide, and and this, and all the way, and so many different drugs. But what can genetic sequencing, identify those proteins which can target for treating COVID? Is there some
Right. So I think understanding the structure, the function, has led to rapid what is called drug repurposing strategies, right?
Mm.
So existing drugs, because anything that's a novel, completely novel drug, would have to go through a lot of safety and the protocols and so on before it can even be thought of as a remedy. But what you can do is repurpose drugs. So existing drugs for some other infections or inflammation
Mm.
get repurposed for and that how you actually pick those drugs is actually using things like the sequence, the structure, the information from that. Um, so many people have used that. In fact, in some ways, you could think of remdesivir and all these, drugs
Yeah.
that actually are coming through that repurposing strategies. And similarly, even um, the interferon strategies that, for example, Biocon has a medicine that was developed for a monoclonal antibody that was developed for psoriasis, and now that's being also tried on COVID. So that is going on. I think that's and the use of AI and the use of to narrow down the search to pick up potential repurposed drugs that could be used on patients where and it depends on the type of response that the patient is having, and the cocktail, they might be given a cocktail of medicines, and the choice of those can be guided by informatics and analysis based on AI. So I think that is definitely one thread that a lot of researchers are following. Very early on, I think the SERB had a call for proposals to Indian research community to come up with drug repurposing strategies, and I know many faculty at IISc and so on had applied, and I think one or two of them have been funded. So there's ongoing work in that direction. Professor Raghavan Vardarajan's group has, of course, been working on a vaccine with Sri Chitra Tirunal, and but you're right that there is strategies that are being used based on sequence and structure to go after therapeutics. But I think the real um, interesting sort of strategy being worked on is also this whole use of neutralizing antibodies, right? And figuring out where how to get to neutralizing antibodies. This convalescent plasma has not worked as well as people were hoping, and so I think more sort of synthetically created antibodies. No, I mean they're still biologics, but they're not necessarily just taken from somebody's serum and given to you, right? There's more sophistication to it. There's a company called Regeneron that has been doing a lot of work on this, and I guess President Trump was given the Regeneron cocktail, that it was pretty remarkable how he bounced back, fairly quickly, right, from an infection, where he was actually put in a very high risk category because of his body mass index and so on. But he something seemed to have worked, right? in his case. And I think maybe it was the Regeneron cocktail. Right? We don't know. I'm sure Regeneron will do some marketing in that at some point. But
That's very interesting to know.
right,
Sir, Shiva here, sir, Shiva Kumar.
right.
Sir, with masks
Mhm.
and social distancing, how bad can India's second wave of COVID can be?
Yeah,
Are there any predictive models?
right, right,
Because winter is approaching and people are fearing the winter.
right, right. So I think masks and social distancing, will always have a very positive impact on keeping the infection spread down. Masks actually, I guess, is to really protect people around you. Um, uh, for protecting yourself, I think you need a very, very tight mask, which is quite uncomfortable. Most people won't be able to tolerate it. Right? I think healthcare workers are forced to use it, but for most people, it's very suffocating. Um, I think the big thing is to keep away from, as I was telling you earlier, I think one of the major factors is the inoculum. So if you're in environments which are closed environments where there's likely to have been a lot of virus let out into the air, there will be micro, droplets and clouds of micro droplets of virus, and you just have to walk through it, and you will get exposed, right? It's so that is the real danger. I think it's these enclosed spaces where there are people spreading infection. Um, I think and in winter that does happen, right? You can't be out, you can't be as ventilated because of the weather, and so there is a greater risk of infection. And certainly, going to malls and movie theaters and restaurants and places like that is a risk. Right? I think, um, I guess I'm also in a very high risk category, so I'm probably a little risk averse here. But I guess that is that's sort of my reading is that we just need to be careful for a while, I think, and if you want to meet people, try to do it outdoors, or if you if someone if it's possible. I mean, it's obviously can't always be the case, or at least have some ventilation going. And just it'll help, because I think the real problem will be these closed spaces.
So you mentioned that we have to live with it for maybe
Sir,
another 1 or 2 years. You think by that time we would have achieved herd immunity?
Yeah, so I had a slide on herd immunity, but I didn't want to get into it. Um, yeah, I think it's a very complicated thing, this herd immunity business, because typically with an R0 of about 2.2, which is what SARS-CoV-2 was analyzed with, or COVID-19 was analyzed with. It looks like you may need to get to about 60% of the population to be infected before you get real herd immunity. Now, there's been some analysis saying that there may be a large segment of the population that is immune, right, for other reasons. Right? They just have, I don't, either some T cell activity, or have been exposed to other infections and therefore have built up some immunity. And so if it is only a section of the population that is really susceptible, then 60% of that smaller population might be a much smaller number than 60% of the whole population. And that argument was made for UK, for example, and why, in some sense, they were able, it sort of subsided on its own suddenly. And but the final word on that is not been settled yet. So herd immunity, how many people have been exposed in India? I think officially we say 8 million or so, but it's probably at least off by a huge factor, right?
So you mean it could be a couple of multiples?
Yeah, the expectation is that it's probably more than a couple of multiples, right?
Okay, so that's scary. Okay.
Probably in the, I mean, I think, it's well known that, for example, cause of mortality, cause of death in India is only reported in 1 out of 6 deaths.
Oh.
Okay. Because, you have to get to the hospital, you have to get some physician to sign off on the cause of death and so on. And that doesn't happen all the time, right? So if you're declaring a certain mortality rate, then, is that the true mortality rate? We don't know. Now, for example, the government's reports right now of recovery rate as having gone very high and so on, that, epidemiologists like Jacob John from CMC Vellore and so on, they argue that you can't really say that, right? You can't say that recovery rate has gone from 60 to 80%. Recovery rate is, in epidemiology, is just those who didn't die, right? Those who don't die eventually recover, right?
Yes.
And so really, if you say 1.9% is the mortality rate, then 98.1% is the recovery rate. So you can't really be talking about increasing recovery rate and so on. That's the so there's some challenges with how things are reported, and what are statistics really meaning and so on. And so I think we all have to become epidemiologists. I think that's the final thing, and it's true. I think lots of people who have background in mathematics and informatics have in some sense become epidemiologists because of this pandemic. So,
Professor Shiva, you had a question. Sorry, I interrupted you.
Sorry, sir, I was trying to know how bad can the second wave in India could be, because now the Europe is suffering from such a wave.
right.
So are we expecting a worse situation than what we currently are facing?
See, I think from a modeling perspective, none of these models are actually that precise that you could say anything.
Yeah, that is exactly why I was asking you.
Right.
I'm not able to see a second peak in any model easily.
So our supermodel guys have, the guys from Dr. Vidyasagar and others, who Manindra Agarwal and so on, they've just written a paper on this. But they're basically saying that if social distancing and so on is maintained, then there's a possibility that this will all die down by February. That was their prediction. The second spike business, I guess very few models will show that. I saw some interesting data on containment zones that you remember in Bangalore city, we had various areas that were sealed off, right? Because they became sort of hotspots. And when you looked at the infection within those containment zones, you had multiple peaks because they would peak, subside, and then suddenly somebody would go over from one building to another building, and then the infection would pick up again, and although the area was contained, there was movement within the containment area, and so you got actually multiple peaks in that data. So there was a whole sort of smart city group that was monitoring some of those containment zones. So in their data, we saw these multiple peaks. So these things can happen in a closed system, right? But when you just model it as with these simple compartment models, you'll not see multiple peaks.
Okay, thank you.
Let me again formally thank you for accepting our invitation. Hope to see you again, probably next edition, and hoping against hope that it's going to be an in-person conference. So,
Right.
so again, thanks a lot, Professor Chandru.
Likewise.